We're talking about this need for research. Of course, what can I talk as a scientist? Need for research. But if we are talking about individual sensitivity, there is well-known and scientifically well-established phenomenon of individual sensitivity. But when we are coming to EMF, somehow scientists are forgetting about it. Individual sensitivity to radiation is well known. For example, for ionizing radiation, for UV radiation, for ultrasound. It is well known. Some studies have shown that only some persons or some animals or some cells will respond to EMF exposures. For example, epidemiological studies showing brain cancer increased risk, only some people in highest exposed group will have increased risk of developing brain cancer. Not all of those people who are most exposed will have this problem. Even better, it is seen in NTP study, where they exposed rats to very high level of radiation. This high level of radiation, the temperature of whole body of rat was increased by 1 degree Celsius. So it was very high exposure. However, out of 90 rats, only 6 got cancer. Others didn't. So not everybody, even with so high exposure, even they were inbred rat strain, developed cancer. Then, of course, many laboratory studies, in vitro exposed cells, also from my lab, have shown that different cell types might have different sensitivity. This is what we have shown, that one endothelial cell line and subset developed from this endothelial cell line, when they were compared, they had different sensitivity to EMF. Different genes were being expressed in response. Last one. So it is scientifically justified to claim that the individual sensitivity exists for EMF exposures. And of course, when we are thinking about self-diagnosis of sensitivity, there are two general lines. That first, that EHS reports, news media affect people's mind, and they are beginning to develop symptoms of EHS, or that health symptoms of EHS appear first. People have unknown health problems. They begin to ask doctors, don't get any help, so they go to Dr. Google and they find. So there are 3 types of scientific studies on EHS: surveys, provocation studies, and biochemical and physiological studies. All of them have one major flaw, namely they have unconfirmed self-diagnosis of EHS. All people who were used in those studies, who were considered as EHS people, they were self-diagnosed. There is no method to diagnose it. So this majority of research data is subjective, so meaning this is not objective information when you ask EHS person, how do you feel? Then is lack of objective markers for EMF effect, so that's why we cannot diagnose it. Of course, there is problem with low numbers of EHS volunteers participating in studies, because in a way, they also are afraid of it and pretty scared that they may die of exposures during experiments. There is a very large diversity in EMF exposure protocols, so this every scientist for himself or herself does something. And acquisition of data is either during exposure or just after exposure, meaning this is only for acute effects, not for delayed chronic effects. And here already Martin has mentioned this study from Schmidtchen and co-workers, and on methodological limitations of experimental studies. And finally, after reviewing some 845 studies, which were just collected by searching different databases, they had only 28 were selected as having sufficiently enough good quality. 21 of them had not shown effects, 7 reported some effects, and there was no any restriction to specific exposure that would be causing it. But of course, in the end, all those studies have this vulnerability that first, they don't know whether their volunteers had really EHS or not. And secondly, there's issue of asking people, do you feel EMF exposure or not? Well, ask me, do I feel ionizing radiation exposure? Ask me, why do I not feel UV exposure, and so on. Why, in this case, EMF, we are using it as a sort of major tool to dismiss the effect because they say they feel, but they, in experiments, they cannot. Well, historically, it is not so simple. EHS people, for many, many years, were neglected by scientists and by doctors. They got very, very frustrated, and they said, they became simply angry and say, I can feel this radiation, you are too stupid to find it out. So scientists began to look for it and use this idea that they can feel this radiation. And we are in this vicious circle. Do you feel radiation? No, so there is no effect. I have published this year a review of some over 230 EHS studies on quality of EHS research. And general idea or final conclusion, there is a number of general drawbacks in the design of all EHS studies that prevents us to make any reasonable conclusions concerning EHS. So first one is this, what I already mentioned. Are volunteers indeed suffering of EHS? Nobody knows. They are coming and they are saying that they have EHS, that they feel it, feel problems and so on. But they might have misdiagnosed, they might be vulnerable or affected by some chemicals in our environment, and there's plenty of chemicals. In the extreme case, none of the self-diagnosed EHS volunteers has EHS. So meaning experiments are performed on 10 or 20 people, none of them has, and the result of study is there is no EHS. And of course, analysis of volunteer groups that are potentially contaminated with non-EHS persons are becoming sort of worthless. Then there are two different types of selection bias. One is by scientists done when they are selecting EHS volunteers, they say you cannot have this or that disease. At this moment, this stage of discovery science, you should not exclude people who might have some other disease besides EHS, because maybe this other disease predisposes them to be EHS. So those people should be taken. They might be analyzed separately, differently, but they should be taken. And also another bias is when volunteers don't volunteer. One thing is, of course, that they are afraid to volunteer because they are afraid of health effects. But there is also another even bigger problem when they first volunteer and then they withdraw because of some reasons, either because they are afraid of radiation and reconsider, or because they don't like research team that does experiments, and over time they learn what opinion this team has about EHS, and they don't want to be involved in this kind of research. There are such examples of teams. Then another thing is about methods of inquiry, that these are psychological provocation studies. So we are exposing people and asking them different questions. Methods were not tested for this purpose, so none of the experimental methods was demonstrated to be able to detect physiological outcomes of EHS, if it exists. There is lack of positive and negative controls. One of the hallmarks of good experiments is that you have a positive and negative controls, that you can say, my methods work. There is no such a thing. And of course, all psychological provocation studies were designed to examine solely acute responses, as I already earlier mentioned. And placebo and nocebo is often spoken about that it is indicating that there is no such a thing, that we are just imagining, or EHS people are just imagining something. But placebo and nocebo also indicate that our mind, when we think, can affect physiology of our body. We can get headache from thinking, we can get increased temperature, some fever from just thinking. Therefore, news on dangers of EMF, some persons experience some symptoms from the news. And this is used as, well, when we show you a scary movie, you get some symptoms, so it means that there is no any problem with EMF exposures, but this is just in your mind. Well, this is nothing new. This is medical students' disease. Any medical student can say, when they are studying different diseases, those students, at least some of them, get sick of this disease just by thinking about symptoms and connecting with behavior of their own body. So it is not any really proof of this, that there is something or there is nothing. And of course, when there are volunteers taken to this kind of experiments, when they are showed them videos which are dangerous, showing dangers of EMF or no dangers of EMF, those are somehow treated by scientists as there is, I don't know, like a virgin. Nobody heard ever of EMF. But every person who is coming has some preconceived opinion about EMF. Either it is dangerous or doesn't care. But they have this preconceived opinion, and this affects how they will react to this video showed them in experiment. And then there are biomarker studies, which have also this largest studies, or most of studies were made by Dominique Belpomme and co-workers in France. I severely criticize those studies. They don't examine effects of EMF on biomarkers. So this is this problem. They say that there are certain biomarkers of EMF that they, or of EHS that they examine, but they don't know where from this person got those biomarkers or responses. And when they don't expose people to EMF to check what is happening with this biomarker, any claim that they are studying EHS is empty claim. Then, of course, this assumption that EMF was the cause of the non-specific EHS symptoms is not sufficient for saying that we are studying EHS. And published experimental data was of very poor quality, and especially claiming statistical significance without providing any evidence. They were just provided in some places asterisks of significance, and that was it. And none of the proposed biomarkers was shown to be really marker of this whatever they were studying, because I don't know whether they were studying EHS or something else. And they also didn't bother even to look at combination of biomarkers, whether they are expressed 2 or 3 or 4 biomarkers in the same person. They didn't look at this kind of thing at all. So time for change and research should be on sensitivity to EMF, not just EHS. Because EHS by itself has sort of, one could say, bad name in scientist circles. And EHS is only a subsection of sensitivity research. It's one part, but not important, but not only. And then sensitivity to EMF in general might lead to a variety of health-related effects in sensitive persons. So if somebody is sensitive, might develop cancer, very few are, fertility problems, neurological effects, etc., etc. So meaning we are not talking that every user of mobile phone should be concerned, but rather we are talking every sensitive user of mobile phone might be concerned that may get some problem. Of course, not every EMF-exposed person will become sensitive or is sensitive, and not every sensitive person will develop the same ailment. Some of them will develop this, some of them will develop that. And of course, uncharted area are co-effects of EMF and other environmental pollutants. For example, what is the role, if any, of pollutants, of those other pollutants in triggering sensitivity to EMF? Don't know at all. So what I would think that we should do in the future, it is project should be combination of provocation and biomarker approach. It should be completely blinded analysis until after provocation and biomarker data of each volunteer are analyzed and combined. So the scientists who are making final decision what is what don't know at all. There should be used self-diagnosed EHS and non-EHS, and they should be not separated. They should be just bundle of people who are being exposed and then looked at. Then the important part is that person is own control. When we start to have few people and they have, we combine them together, whether some marker goes up or down on average, it is not okay. We have shown this in our experiments on skin of people, volunteers, that this using same person as own control can provide information. And of course, samples of for biomarker analysis should be taken before and after exposure. And in first place is sort of shotgun approach. Samples taken within 1 hour after exposure, later on at other time points might be. And I will explain in a moment why. And metabolites, protein, and gene screening with protein chips and gene chips. Most important, in my opinion, are proteins, because our cells don't change their physiology as long as proteins are not changed inside them. And why I'm saying that 1 hour, within 1 hour, we were doing experiments on cells and on humans where we were exposing them for just 1 hour, and also others have seen activation of stress response, which is very rapid activation. It can be noticed within minutes, and after 1 hour, it is beginning to vanish. So this is thing that if there is any sensitivity effect, first what we should see in exposed humans activation of stress response, bigger or smaller. And this was what we were doing with human skin when volunteers were exposed and they served as their own control, and only after analyzing data and the expression of proteins in single person, then they were compared, those 10 persons, and it was possible within 1 hour to observe small changes in protein expression in our setup. So we confirmed that proteomics screening approach can identify protein targets of RF exposure. So in conclusion, EHS research that has been so far published is of insufficient quality to prove or to disprove causality link. Continuation of psychological provocation studies will not resolve this problem. Biomarkers of sensitivity are needed for determining prevalence, for diagnosis, and for possible pharmacological treatment if we like later on to give them a pill. Studies combining provocation and biomarkers are the way forward, but of course they should have large number of volunteers, sensitive and non-sensitive, and high throughput screening of metabolites, proteins, and genes to find biomarkers. We don't know what we are looking for, we are just looking. And finally, last thing, that this deployment of 5G might worsen exposure situation for sensitive persons, and they are very much afraid and asking questions to which we as a scientist don't have real reliable answers. Thank you.